Morning, everyone.
Morning,
Good
ma'am.
morning, ma'am.
Morning, ma'am.
Today's topic will be on the morphology of bacteria. Okay. Uh, so, have you ever heard of prokaryotes and eukaryotes in your life?
Yes.
Yes, ma'am.
Yes. Okay. Can you name a eukaryote? Can you give an example of a eukaryote?
We human being.
We human being. Okay. Good. Anyone who can tell me or who can give an example of prokaryote?
Ma'am,
Bacteria.
bacteria.
Okay. Okay. So, in the introduction, what are these microorganisms? As I have told you yesterday also. So these are a living structure which can be seen or which can be seen under the microscope. Okay. And these are before, while doing the classification of this organism, they classified into only two kingdoms. We call it as kingdom, animals and the plant kingdom. But later on what happens is it was unsatisfactory. There were certain things or certain organisms which doesn't fall under this animal or in the plant, uh, like you know uh, plant groups. So they coined another term which we call it as protista. Okay, it's known as protista. So, okay, just a minute. Let me present the slide. After that, we'll start. Is it visible? The color is visible?
Yes, ma'am.
Yes, ma'am.
Okay,
Yes, ma'am.
fine. Let me. So these organisms, as I have told you, these are living structures of microscopical size, very small size, which cannot be seen with your naked eye. We need an aid. For example, we need microscope to see the organisms. Okay, and yeah, I told you a third kingdom the scientists have introduced, that is known as protista. Under this protista, they have divided into two categories again. What are the two categories? Prokaryotes and the eukaryotes. So some of the examples of the eukaryotes are, you know, fungi, okay, algae, then protozoa, slime mold. You should know these names, okay? What are they? Fungi, algae, protozoa. Protozoa are the ones which we call it as parasites. You will get to know. Have you heard of this thing? Roundworm, hookworm. ऐसा कभी सुना है आप लोग ने?
Yes. Yes, ma'am.
Yes.
Yes, ma'am.
Okay. So protozoa, amoeba सुना है आप लोग ने?
Yes.
Yes.
Yes, ma'am.
So
Ma'am.
Yes, ma'am.
amoeba is one of the example of protozoa, and this uh, what is that? What did I say? Ringworm, sorry, roundworm, then this one, hookworm. This comes under parasites. When we study parasitology later, some of the parasitology groups, topics are also there. Slowly, I'll let you know. Okay. So, fungi, algae, protozoa, slime mold, these are the ones which comes under eukaryotes. And this one, prokaryotes like bacteria and the blue-green algae. Okay, bacteria and the blue-green algae are the examples of prokaryotes. Now the next slide. Here we are going to discuss the certain differences between a prokaryote and eukaryote. Okay, you can zoom your slide. Okay, you must be knowing, I guess. You can zoom it out.
Ma'am.
So, um, so prokaryotes and the eukaryote, there are lots of differences. The first differences is the grouping of these organisms, as I have told you. Under the prokaryotes, will have bacteria as well as blue-green algae, whereas for the eukaryotes, we have fungi, parasites, and other algae, plants, and animals. Okay. Now, the presence of the nucleus here in the prokaryotes, it is diffused, means it is not well defined, whereas in the case of eukaryotes, it is quite well defined. It has got its own shape of the nucleus has its own shape, whereas it is not there in case of prokaryotes. So we call it as diffuse nucleus. Again, the nuclear membrane is present or not. Okay, so nuclear membrane is absent in the case of prokaryotes, whereas it is present in the case of eukaryotes. Okay, so I might ask in the viva, is nuclear membrane present in the case of prokaryotes or otherwise bacteria? You will say no. Okay, whereas in the eukaryotes it is present. Then nucleolus. Nucleolus is also present in the nucleus actually. So nucleolus, it is absent in the case of prokaryotes, whereas it is present in the case of eukaryotes. The third one, that is ribonucleoprotein, RNA. That is in the prokaryotes, it is absent. Mostly bacteria, they have DNA. Okay. Then eukaryotes, it is present. Ribonucleoproteins are present. Then cell division in the prokaryote is by the method which is known as binary fission. Whereas in the eukaryote, the method of reproduction is or cell division is by mitosis or meiosis. Okay. Then the chromosome which are present in the case of prokaryotes, it is only one and it is circular in nature. Whereas in the eukaryotes, the chromosome used to be many and it is linear shape. Okay. Now coming to the third category, first major groups हो गया, nucleus हो गया. These things which I was explaining about nuclear membrane, nucleolus, ribonucleoprotein, cell division, chromosome, this comes under nucleus. The third one is extrachromosomal DNA. Okay, extrachromosomal DNA, it's a kind, you should know from today when I say extrachromosomal DNA, you should not confuse. Let me explain you what is extrachromosomal DNA. There are certain DNA which are not present in the nucleus. There are certain DNA which are present outside the nucleus, that is in cytoplasm. Cytoplasm के अंदर nucleus present हो रहा है कि नहीं? Yes. So the nucleus, the DNA which is present inside the nucleus, we call it as chromosomal DNA. Whereas the DNA which is present outside the nucleus and which is present, that means it is present in the cytoplasm, that is known as extrachromosomal DNA. And ये किस में found करते हैं? It is found in the plasmids. ये prokaryotes के अंदर होता है. That means let's say an example, that is bacteria, which is a prokaryote. It has got a structure which is known as plasmid, and this plasmid is the extrachromosomal DNA which is present in the cytoplasm. So in short, you can say plasmids are present in the case of prokaryotes. Okay. Then eukaryotes में the extrachromosomal DNA are found in the mitochondria. Okay, it is found in the mitochondria, which is also known as the powerhouse of the energy. Okay. Now coming to the cell membrane. The cell membrane of the prokaryotes doesn't contain sterols. Sterol is a kind of a lipid, okay, except in the mycoplasma. Mycoplasma करके एक bacteria है Okay, mycoplasma करके एक bacteria है. Except in that bacteria, others में does not contain sterol. Whereas eukaryotes के cell membrane में it contains sterols. Okay. Then cellular organelles like mitochondria, for example, in the prokaryotes, that is bacteria, they are absent. Okay, mitochondria is absent in the case of prokaryotes, whereas it is present in the case of eukaryotes. Now coming to the ribosomes, it has it is made up of different units. Ecto 80S unit, another one is 70S unit. So in the case of bacteria, the ribosomal unit is 70S, whereas in the case of eukaryote, it is 80S. Okay. Then respiration कहाँ पे हो रहा है? In the bacteria, the respiration is taken place in a structure which is known as mesosomes, okay, which is present inside a bacteria. Whereas in case of whereas in the case of eukaryotes, the site of respiration will be mitochondria. Okay. Then pinocytosis. Pinocytosis is a kind of process, okay, where cell eating, उसको भी बोलते हैं cell eating, where cell used to engulf, okay, certain things. That is pinocytosis. It is present in the prokaryotes, whereas it is absent in the eukaryotes. So these are the major differences between prokaryotes and eukaryotes. You can also find out by yourself whether other than this, what there are lots of different differences between the two. You can find it out and write by yourself. Okay. The third slide will show, yeah, the size. Coming to the size of the bacteria. The bacteria is quite small, as we have as I have been saying. It is the size of the bacteria is not mainly, it is measured in a unit which is known as micron or micrometer. So it is denoted by this this micrometer. Okay, this way. So the medically important bacteria, that means medically important bacteria का मतलब है the ones which are involved in causing any diseases or any infection to human. Okay, the sizes ranges from 2.5 micrometer length in 2.2 to 1.5 micrometer width. Okay, you should, if you can, you should be able to remember this particular size. So in the Viva, I can ask you, what is the size of a medically important bacteria? You should say 2 to 5 micrometer in length, 2.2 to 1.5 micrometer in width. Okay. So this bacteria, as you can't see with the help of as you can't see with the naked eyes, we have to see through something. So we use microscope, and different kinds of microscope we use. These are the different microscopes which we use. Bright field or light microscope, which we use in the laboratory. Okay. Dark field microscope, phase contrast microscope, fluorescence microscope, electron microscope. So we'll be dealing with the light microscope. These are all dark field, phase contrast. Dark field में भी क्या होता है? The organism will will be illuminated. For example, you are going to see bacteria. So under the background will be dark, whereas the bacteria will be illuminating. Okay, it brightens. So that's how we get to know it's a bacteria. Then phase contrast microscope. In this kind of microscope, what we use is the contrast of the contrast created by the sizes of the or by the structures, different structures which are present inside the bacteria, whether it can be mitochondria, okay, whether it can be mesosomes, whether it can be something else. Okay, so they have their particular contrast, and we use a different variety of condensers for this kind of microscopes. Okay, then we don't have this in our this microscopes we don't have. Then on the only one which we'll be focusing is on the light microscope, which are we using in the laboratories and all. Okay, in normal laboratories. These are mainly for the research purposes and all this one. Fluorescence microscope. This is also a kind of microscope where we use certain dyes, fluorescent dyes we call it. Yeah, like a particular dye we use to dye the organisms. Then after that, the organism will flourish. It will show certain colors. And then electron microscope. This is also like here in the electron microscope. See, by the light microscope, we cannot see the nucleus and all. It's quite minute. So for seeing the nucleus and all, we need electron microscopy. This is a more, it has more resolution power compared to the light and all. Okay, so this is the structure of the microscope, which is like yeah. So the light microscope which we use in the laboratory, it can be either monocular or otherwise it can be binocular. Okay, so in the monocular microscope, we the eyepiece will be only one, whereas in the binocular we have two eyepieces in that. Okay, so this one is monocular. This part, the microscope is divided into three parts. Number first, this portion is the head portion. This portion is the arm, and this is the base. There are different parts, different names are given to different parts. This portion, the first portion, we call it as eyepiece. A lens will be here in this eyepiece too. Okay. Then this is the tube. Eyepiece tube, we call it as the eyepiece tube. And yeah, this one. This one, this particular part is known as the nose piece where the objective lens will be fixed. So the objective lens, uh, it can be the lowest objective lens, the power is 10x. Okay, then the high power will be 40x. Then another one is the oil immersion where like we use 100x for that. Okay, most of the staining where staining procedure is used. For example, you must have heard of gram staining and all. This is in the microbiology, the word gram staining will be very much like used. Okay, so in most of the staining parts where you stain the organisms, we will be using the 100x. And for seeing just the morphology and for seeing the motility and all, we use 10x or otherwise we use 40x or 45x. Okay. So this is the objective lens. So either it can be 10x or it can be 40 or 45x or 100x. And the eyepiece has a power of 10x. So when someone asks you what is the resolving power, okay, there is a word which is known as resolving power. Actually, resolving power, it is known as the ability of the lens to clarify or to separate uh, two objects from each other. Okay, so the power we have, how will you calculate the resolving power is, for example, eyepiece ka resolving the eyepiece ka lens is 10x, and let's say the objective lens ka 10x. So what is the resolving power? What we do is what we know is this particular lens will magnify the object 100 times. So 10 into 10 is equal to 100. Okay, so we are going to see whatever your eyes will be able to see, that is 100 times magnification of that particular object. For example, again, when 10 eyepiece is 10x and you are using 40x lens. Okay, 40x. So the magnification power which you're going to see by your own eyes is you're magnifying the object. It can be bacteria, let's say. You're going to magnify that bacteria into 400 times. from its own size. Then if the eyepiece is 10x and if you're using 100x, so that means you're going to magnify the particular object, it can be bacteria, it can be anything else, into 1,000 times its shape or its size. Okay, got it? And this part, it has got a clip. There are two clips here. If you are able to see it, you can zoom it out. There are two clips. This is for fixing the slide. Okay, so that the slide doesn't move here and there. We call it as stage clip. Okay, so it has got two stage clip, and this particular area is known as stage. This platform, black color platform, is known as stage. And there is a structure which is present here. We call it as a condenser. Okay, condenser. Then we have a structure here. This part we call it as a iris diaphragm. Okay, so iris diaphragm, and there is a hook where you can control the light. Okay, where when you focus at the 10x, the iris diaphragm should be totally open. When you focus at 45x, sorry, in the 10x, the opening of the iris diaphragm will be lesser. Whereas in the 45x, you have to open it in a, like, in a medium way. Whereas in the 100x, it should be totally open. Okay. And there is this one is the on off switch. This is the base, which I have already said. Yes, this is, we call this as nope. Okay, nope. KNOB, nope. So we can use this knob to bring the objective lens up, down, left, right, or right, left. Okay. Then another knob is there which is used for fine adjustment, where after you focus, you use this particular knob to clarify the image. Okay, for clearing the image. We call it as fine adjustment. And this one is known as coarse adjustment. Okay, coarse adjustment knob, and this one is fine adjustment knob. And this one is arms. So these are some of the structures. What you can do is, you draw a microscope, you maintain a practical book also. You buy a practical copy, start drawing the first you draw this
No, sound is
this
not.
level, and you is it clear?
Ma'am, you are not audible.
Is it audible now?
Yeah, now audible.
Okay, okay. So what I was saying is, you when you go to market now, I think the markets are open only. You buy a notebook, maintain it for the practical also. Side by side with the theory, I'll be telling you what you should do in the practical. Okay. The first practical will be on light microscope only. Microscopy. So you will draw this microscope. It is there in the Google only. You draw this, level it properly, and each part you have to write the function. For example, ऑब्जेक्टिव लेंस का क्या फंक्शन है, कंडेंसर का क्या फंक्शन है, डायफ्राम का क्या फंक्शन है, you have to write it. Okay. Then one more thing, on the first page, before the light microscope, in the first page, I want you all to write the do's and the don'ts in the laboratory. What you should do and what you shouldn't do in the laboratory. This can be found in the Google only. Okay. So study it, understand it, and jot it down or write it down in your practical file on the first page. Are you
And
understanding?
do and don't.
Yeah, do's and don'ts, what you should do in the laboratory and what you shouldn't be doing in the laboratory. Okay, that is
So,
not a experiment number one. It is just for your knowledge. That will be the page one. And coming to the second page, what you can do is experiment number one, light microscope. Diagram, you level it properly, then write the functions of this, all the parts of the microscope. Understood? Okay.
Yes, ma'am.
I'll take it.
Yes, ma'am.
Okay. You can. Now, yes, coming to the shape of the bacteria. Okay. I think this is more visible or pehla wala ठीक था?
This is visible.
Is it visible? Okay. There are lots of varieties of organisms. There will be lots and lots of bacteria. There will be lots of viruses. They have different shapes. They have different structures. Okay. So here, as we are dealing with the bacteria for now, let's talk about the bacteria. According to the shape, okay, according to the shape, the bacteria can be divided into different varieties. Okay, I'll take the slide. See this. These are the different types of bacteria. Okay, slowly, let me explain this first. The first one we call it as cocci. How do you pronounce it? Cocci. Okay, cocci. So cocci is the singular word. When you say cocci, it means coccus. Okay, and then it the name is taken from cocos, meaning berry. Berry is a kind of like this thing. Gu, I don't know. I forgot the name of those berries. But let's take an example of grapes. Grapes ka round shape with the bundles. Okay, it used to be in the bunch. You remember that grapes का how people how those दुकानदार sell करते हैं पूरा उसको bunch में sell करते हैं ऐसा structure है ये कोकाई. So these are the bacteria which has a oval or spherical in shape. They have a shape of oval or otherwise spherical. For example, this one. Can you see this? These are spherical shape and they are appearing in clusters or we call it as groups. Okay, so one circular thing is one bacteria. Keep in mind, one circular thing is one bacteria. So lots of bacteria is like making this particular shape. Okay, so we called it as a cluster. Till here, you are understanding the first shape of the bacteria is cocci. Cocci is the when I ask you what is a cocci, what is the shape of a cocci? It is oval or spherical in shape. Understood?
Yes, ma'am.
If you are not understanding, let me know. Till here, are you understanding?
Yes,
Yes,
ma'am.
ma'am.
Yes, ma'am.
Yes, ma'am.
Okay.
Ma'am.
And what are these clusters pair?
I haven't gone that yet. Okay, I'll
Oh,
explain.
okay, spherical. Okay, I think
So here, up to here, I have explained cocci means it is oval or spherical in shape cells. ठीक है. Again, cocci is again classified into different varieties. One is which are found in clusters, groups. जैसे कि grapes का bunch होता है, अंगूर का जो bunch होता है, ऐसे ऐसे दिखता है. For example, this one which I was showing. This is known as cluster. Fine. So an example of a bacteria which is a cocci and which appears in the cluster is Staphylococci. You must have heard before Staphylococcus aureus. Okay. The name of the bacteria is a little complicated, but slowly you'll get it. Okay. For now, just know that a cocci which appears uh, in the cluster. Okay, this is known as cluster. An example is staphylococci. Now coming to the first slide again. Cluster I have explained. How does it look like? You can add here one point, grape like clusters or grape like shape. Okay, जो अंगूर का bunch होता है. Now coming to pair. So this cocci can be appearing as a pair, means do. That kind of structure is known as diplococci. This one, two. So if they are appearing in this way, we call it as a diplococci. They are appearing in pairs. So example of a diplococci is Streptococcus pneumoniae. Okay. Number third one, cocci can also be arranged in the chains. Okay. Cocci can also be arranged in chain. So chains may, if it appears this way, let me show you. This one. एक के बाद पूरा चेन बनाते हैं ये. This kind of
No, it's like
structure,
artworm.
it's not like artworm. It's because of the structure, you know, how they have like made it. Otherwise, okay, let me show you. I think it's not here. Uh, Tomorrow I'll show you the actual one. This is just a presentation what they have done. So round karke, it will be in a short chains or it can be in the long chains, and we call it as streptococci. So an example of a bacteria which is a cocci and which appears or whose arrangement is in chain is Streptococcus pyogen, or you can just say streptococci. Okay. The third one, groups of four. So cocci can also be arranged in four groups. We call it as tetrad. And see here. One, two, three, four. As a char. Okay. An example of a tetrad is micrococci. यहाँ पे लिखा ही नहीं उन लोग ने. So an example of a tetrad is micrococci. Then we have a group of eight. So cocci can also be grouped in the group of eight. Example is Sarcenia, ऐसे दिखता है group of eight. When you see, these are all seen under microscope. Okay, naked eye नहीं, microscope में दिख रहा है ये सब इसका arrangement. Sarcenia, see this. Group of eight. 1, 2, 3, 4, 5, 6, 7, 8 at the back. 3D structure है ये. Okay. यहाँ तक समझ में आया आप लोग को?
Yes, ma'am.
Yes.
Okay.
Yes, ma'am.
So what is cocci now? What is a cocci?
Bacteria which are spherical in shape.
Yes, a bacteria which is spherical in shape. And what are the possible arrangement of this cocci?
Clusters, chains,
Clusters.
and
Chains.
Yeah.
Pairs, then group of four or
Chains.
group of eight. Okay. So you should know, there are microbiology is a very vast chapter. Okay. Lots of things are there. Lots of things can be asked as a Viva question. So you have to learn by heart all this. I can ask, can you name some organisms which are cocci in shape, which are spherical in shape, or which are cocci? So you should know the names of those organisms. You have to list it out and keep for yourself. Okay. So this is about cocci. The second shape is, where is it? Bacilli. So bacilli is a rod shape actually. So you can add rod shape. Do you know what is a rod?
Yes.
So this kind. Rod shape, rod. Yeah, this kind, elongated one, elongated type. Okay, these are known as bacilli. So in just a minute, okay? Hello. So what is that? Where was I? There is always interruption.
Road
Okay,
shape.
bacilli. Okay. So the bacilli, it is rod shape or otherwise it can be curved shape. Okay, it has थोड़ा सा curved shape ऐसे होता है और otherwise sometimes some bacteria shows coma shape. ठीक है, कोमा जैसे कोमा नहीं है, exclamatory कोमा ऐसे. Okay, यहाँ पे this kind. थोड़ा सा कोमा जैसे लगता है ना. So this kind. This is known as coma shape. Then another one is cuneiform shape. It has got a pattern. Okay, cuneiform ka pattern. जैसे later I'll show you what is cuneiform and all. So here again this bacilli, उसका arrangement बहुत सारा arrangement होता है उसका. One is cocobacilli. So it looks little bit like coccus also. It looks little bit like bacilli also. मतलब it looks little bit circular. It looks little bit like rod shape. So it's a short rods we call it. For example is brucella. Example of a cocobacilli is brucella. Brucella is the name of a bacteria. Okay. यहाँ पे है कि नहीं है cocobacilli? Okay. See, this example, I was talking about cocci, no? These are known as cluster form. This is Staphylococcus aureus. Okay. This is how the tetrad looks like. And this is bacilli. This is bacilli. Let me see in this what I was saying. Yeah, comma shape, this one, comma shape a bit. Cocobacilli, yeah, somewhat this one. It's a very short, it's a very short, like, it looks little bit of cocci as well as bacilli. And this is cocobacilli. The actual bacilli has to be this way under the microscope. Okay. But when it is cocobacilli, it looks little bit short like this. Okay. Later, when like धीरे धीरे पता चल जाएगा. Just let's go through this once. One is the second one is we call it as streptobacilli, where bacillus are arranged in the chain. Okay. ये वाला. Streptobacilli. It looks like chain. Strepto means chain. Please keep in mind. Strepto का मतलब है chain. So this is chain and streptobacilli. Okay. So example may you can write just streptobacillus only. Then third one, Chinese letter or cuneiform pattern. Here we were discussing about a cuneiform pattern. It has got a pattern. I'll show you how does it look like. This one. It looks like cuneiform pattern. This is the pattern where how to show. It has got an angle actually. Okay, it has got an angle. You must have heard of Corynebacterium diphtheriae. कभी सुना है जो diphtheria cause करते हैं? So that particular bacteria has a cuneiform arrangement where it has got angles. Okay, angles are made in the bacteria in its body. ठीक है? Then next is comma shape, which I have already explained. Spirilla. Spirilla is something which is like in a spiral form में होता है. ये वाला. थोड़ा सा spiral shape जैसे होता है. Okay, this is known as spirilla. Examples will be spirochetes. Okay. So this is about bacilli. Then we have this thing. The yeah, gram staining property पे आ गया अभी. So understood the shape of bacteria when I asked you what are the shapes of bacteria, you can say it is broadly divided into cocci and bacilli. Okay. It is broadly divided into cocci and bacilli. Again, in cocci, it is divided into different ways, different arrangement. The first arrangement is cocci are arranged in cluster. That means grape-like structure. अंगूर की तरह होता है, वो bunch में form होता है. Second one is pair में होता है. So cocci which are arranged as two. Okay, that is also known as diplococci. Third is when they are arranged in chain. Okay, when the arrangement of the coccus or cocci is in chains. Example is your streptococcus. Another one is it can also be arranged as a group in the group of four, that is known as tetrad. An example of tetrad here I haven't mentioned here. You can write micrococci. Okay. Then group of eight, it's Sarcenia. Okay, this one. Let's see this once again. Two means diplococci. This one is chain, that's why streptococci. This is cluster form of cocci. So example is staphylococci. This is in the group of four, we call it as tetrad. An example is micrococci. And group of eight, an example is Sarcenia. Understood? Then for the bacillus can be Coccobacillus, means it has the shape of a coccus also as well as bacilli also. So it will be a short kind of rods. Okay. Then second form is यहाँ पे नहीं है actually Coccobacilli का structure यहाँ पे नहीं है. The second one is Streptobacilli, where bacilli are arranged in chains this way. Example is Bacillus anthracis. Then the third one is what? Chinese or cuneiform shape. Okay. It has got a shape which is angular in nature. Uh, I'll show you tomorrow how does it look like. But when you this thing gram staining करने के बाद ऐसे दिखता है ये cuneiform shape angle पे. Okay. And यहाँ पे आप लोग dots दिखाई दे रहे हो यहाँ पे ends पे. These are a place where they store ATPs. They store energies. Okay, for which is used for their survival. Okay. अब कहाँ चला गया? Okay, fine. Now is comma shape. It has got a comma shape. Comma shape ये हो सकता है. This is comma shape. Example is Vibrio. कभी सुना है Vibrio cholerae जो cholera cause करता है?
Yes.
Yes, that particular bacteria has a shape of comma, like this. Okay. Then spirella. Spirella, it's in the form of spiral. जैसे स्पाइरल बाइंडिंग होता है ना, जैसे कि तुम लोग का नोटबुक में स्पाइरल बाइंडिंग होता है. तो स्पाइरल का मतलब है कर्व्ड और दिस काइंड ऑफ शेप, ये वाला. ये ठीक है. Any questions till now up till this? Understanding?
I have some confusion.
क्या confusion है सैन भी बताओ.
Spiral में.
स्पाइरल में क्या confusion है बताओ. Spiral, these are the bacteria. Okay, these are bacilli, means rod shaped structures है bacilli. ठीक है. हमेशा याद रखो bacilli का मतलब है rod shape, cocci का मतलब है round और spherical shape. ये दोनों चीज याद रखो पहले. ठीक है. उसके बाद ये spiral shape bacilli में आता है and it has the shape of this, ऐसे curve वाला shape हो रहा है. दिखाई दे रहा है ये part? Are you seeing on your screen? ये Sandeep. आपको दिखाई
Yes,
दे
ma'am.
रहे हैं?
हाँ हाँ दिखाई
ये
दे रहा है।
शेप को बोलते हैं स्पाइरल। समझ रहे हैं आप? माइक्रोस्कोप के अंदर दिखता है ये। ठीक है।
हाँ spiral shape.
एग्जाम्पल है ट्रेपोनेमा पैलिडम। आपने सुना होगा सिफिलिस। यू गाइस हैव हर्ड ऑफ सिफिलिस?
Yes.
ma'am.
This is a sexually transmitted disease. Okay, it is caused by Treponema pallidum and उसका shape है spiral shape. ऐसे दिखता है माइक्रोस्कोप के अंदर। ठीक है। और ये बहुत काफी डेलिकेट ऑर्गेनिज्म है। वो भी ऐसे ऐसे वैसे दिखाई नहीं देते। उसके लिए अलग से स्टेनिंग प्रोसीजर होता है। वी कॉल इट एज इम्प्रेग्नेशन स्टेनिंग। ओके, इम्प्रेग्नेशन मेथड, जहाँ पे हम लोग कुछ और डाइज लगा के उसको फिक्स करते हैं। उसके बाद ही दिखाई देगा उसका स्पाइरल शेप। ओके, यहाँ तक आप लोग टुडे क्या क्या पढ़ाया मैंने? माइक्रो ऑर्गेनिजम्स का डिवीजन पढ़ाया, देन प्रोकैरियोट्स यूकैरियोट्स के बारे में पढ़ाया आप लोग को, डिफरेंशिएशन, देन बैक्टीरिया के बारे में, कौन सा लाइट माइक्रोस्कोप के बारे में थोड़ा बताया, देन उसके बाद स्ट्रक्चर ऑफ माइक्रोस्कोप, है ना, देन शेप ऑफ बैक्टीरिया पढ़ाया आज। ऐसे यहाँ तक करते हैं। ठीक है, यू डू द रिविजन टुडे एंड आई विल गिव यू दिस स्लाइड्स आफ्टर द क्लास। अभी बहुत सारे स्लाइड इसमें आगे है। ठीक है, आई हैव गॉट 24 स्लाइड्स इन दिस। So let me complete the chapter first, and after that I'll share you the slides. For time being, you just sometimes when I'm teaching, you note it down. Sometimes मैं जो बोलती हूँ वो यहाँ पे स्क्रीन पे नहीं होता, या अपने लेकिन मुंह पे बोलते हैं. So you just note it down. Important. You have the habit of keeping your eyes and ears open, and always like your hands to be ready with a pen. So extra जो मैं बोलती हूँ कभी कभी वो note it down करो, because it will help in your viva. समझ रहे हो आप लोग?
Yes, sir.
And one more thing, have the habit of reading by yourself sometimes, because teachers like, okay, they will give you the slide, but still एक होता है, in my previous colleges and all, never our teachers never gave notes. We have to make our own notes. We have to study by our own. Here we are spoon feeding you all. Okay, so have the habit of making your own notes. That will your hard work will be seen and you'll score more. Okay, anyways, let me take the attendance. Geometry Brahma.
Yes, ma'am.
Lanchen B. Shanti Bala.
Yes, ma'am.
Sandeep.
Present, ma'am.
Sudeepa.
Yes, ma'am.
Sudeepa. Malvika.
Yes, ma'am. Yes, ma'am.
Joshila.
Yes, miss.
Okay. Then Joshila is present. Fine. Saikat.
Yes, ma'am.
Nikita.
Present, ma'am.
Arpita. Arpita is present.
Yes, ma'am.
Akash. Akash is present. Absent. Pooja Devnath.
Yes, ma'am.
Pooja is present. Okay.
Yes, ma'am.
Prakash.
Yes, ma'am.
Abhijit. Abhijit Das. Priyasur. Lipika.
Yes, ma'am.
Caroline.
Present, ma'am.
Arga Jyoti. Arga Jyoti. Russell.
Present, miss.
Dishant.
Yes, ma'am.
Sagar.
Yes, ma'am.
12. 12 was
Present, ma'am.
present, huh? Okay. Vishwabandhu,
Yes, ma'am.
Vishwabandhu is absent. Sonia,
Present, ma'am.
Blikania, Bojita,
Present, miss. Yes, ma'am.
Sohina,
Yes, ma'am.
Lanchenba,
Present, ma'am.
Galaxy,
Present, ma'am.
Jessica,
Present, ma'am.
Ramrigam,
Present, ma'am.
Manika,
Yes, ma'am.
Amus,
Ma'am.
Parvati. Parvati was present.
Present, ma'am.
Huh. Bakri,
Yes, ma'am.
Aprajita. Okay, okay. Tennyson,
Yes, ma'am.
Kosik,
Yes, ma'am.
Ishika,
Yes, ma'am.
Tafikul,
Present, ma'am.
Deepa, Deepa Sutradhar, Lipokumla, Lipokumla,
Present, miss.
Satarupa,
Present, ma'am.
Dipjoy,
Yes, ma'am. Present.
Dipjoy, Subandu,
Present, ma'am.
Yes, ma'am.
Subandu,
Yes, ma'am.
Saurav, Saurav, Lintoinganbi,
Yes, ma'am.
Veshwalu, Livitole.
Present, miss. Present, miss.
Yeah. Cello, Cello is present, absent. Anisha,
Present, ma'am.
Rakesh,
Present, ma'am.
Priya,
Present, ma'am.
Arindam, Pushpashri,
Present, ma'am.
Asha Begum,
Present, ma'am.
Reena Deburma,
Present, ma'am.
Rahul Kapali. Okay. Thank you, everyone. I'll send you the link if there is class tomorrow. Okay. So do the
Ma'am.
revision once again. Hanji.
मैम मेरा दिया क्या प्रेजेंट प्रेजेंट
हाँ. Yeah. Okay. Thank you, everyone.
Thank you, ma'am.
Thank you, ma'am.
Thank you,
Thank you,
ma'am.
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 Thank you, ma'am.